So for chapter 2, we're going to look at the central dogma of molecular biology, and the bigger objective that I've designed for this one is to predict how scientists use the features and properties of DNA for discovery. So instead of just saying, these are the things that you need to know, it's how do we actually use those things that you need to know? So we're going to apply it a little bit for this chapter. And so to start, I want you to think about the important features and properties of DNA. And again, this is the handout for chapter 2 that's in the handouts folder. And we actually already did a little bit of this in the first day of class when we looked at the models and you looked at the DNA and you remembered from other classes what are the important parts of DNA. And so this is just a quick reminder. So it shouldn't take too long. When we think about the important parts of DNA, we remember that we have the 4 different nucleotides. We have the adenine, thymine, guanine, and cytosine. So we've got 4. And the pairings for those are A and T and G and C. So A and T go together and G and C go together. The way I remember this is that it's not necessarily the best one, but the skinny letters go together and the rounder letters go together. And another way that I remember another important part of it is that there are 2 bonds between A and T because they're skinnier. And then there are 3 bonds between G and C because they are more rotund or rounder. So they need more bonds to hold together. That's just based on the letters, not based on the actual chemistry of them. And another thing that we need to remember is that there are different types of bonds. And so the quick quiz question is, what is the bond that is between 2 nucleotides within one strand? So that would be this one here. Also this one right here. So what is the name of the bond of nucleotides within one strand? And one thing to remember is that you see the phosphate backbone here. That's the bond that we're talking about. And so we can remember that that is the phosphodiester bond. So phosphodiester bond for nucleotides within one strand. And then the easy cheat for if you're looking at an image and you see the dots between 2 strands, so nucleotides between 2 strands, those are hydrogen bonds. So 3 dots generally represents a hydrogen bond. So we've got A, T, G, and C are the nucleotides, phosphodiester bonds connecting nucleotides within one strand, hydrogen bonds connecting nucleotides between 2 strands, and also the connection between 2 strands is not like this. If you think about your hands as the 5' and your elbows as the 3', the 5 and 5' don't go together. They line up anti-parallel. And we will look at that a little bit in more detail a little bit later about why that is. But they line up anti-parallel, 5' to 3'. And one way to do that is just if you're going to put your arms together, you're going to do this. So you just have to remember that. And if you try it like this, it torques your body in a weird way and it doesn't feel right. So you just shake it off and then you go to the one that actually feels good. The body position that feels good. And if you need to do that when you're taking exams, you can be say, oh yeah, 5' to 3', 5' to 3'. Which also brings up that there is 2 different ends to the strands. So there's a 5' for each strand and a 3' for each strand. And then those are going to line up anti-parallel. Let me see if I got all the things. I probably missed something. So let's go back to, again, also this is in the PowerPoint too. So all these are listed. Oh, right. The one thing I forgot, I mentioned it, but there is a sugar phosphate backbone. So this blue ribbon here is the sugar phosphate backbone. And then the nitrogenous bases are in the middle. And because that sugar phosphate backbone has a negative charge, it is more likely wants to interact with water. Whereas the nitrogenous bases are neutral, so those are going to be lining up in the center that doesn't want to interact with water. When we think about our cells, there's a lot of water in our cells. So the positive, negative charged aspects of molecules and the neutral aspects of molecules are going to determine how they're going to be in the presence of water. So that's actually a really important concept. So we've got built from nucleotide monomers, A, T, G, and C, directional 5' to 3' that lines up anti-parallel. The A and T go together and G and C go together. Phosphodiester bonds within one strand, hydrogen bonds between two strands. And I think that is it. So the important features and properties of DNA. Hopefully this is a little bit of a review for you from some of your other classes. And just as a reminder, we did cover this a little bit when we were talking about the different models that we looked at during the first day of class.